Good morning. My name is Eliana Buenaventura. I am a Peter Buck postdoctoral fellow working with Torsten Dicko in the Department of Entomology. I study flesh flies of the family Sarcophagidae, which belong to the superfamily Oestroidea. Sarcophagidae is divided into these three subfamilies, with Sarcophagidae as the largest and more diverse one. The key on the relevance of the flesh flies is their diversity of life habits. These flies can parasite invertebrates, including insects, and they also feed on carrion and dung. In urban ecosystems, carrion include human corpses. When it comes to crime investigation, usually blowflies are considered as the most important indicators of the time of death. However, in recent studies found that sarcophagids are more diverse than blowflies, and in some cases they are also more abundant in human cadavers. So flesh flies have a great potential as forensic indicators. In wild ecosystems, flesh flies are also more diverse than blowflies, and they constitute the major guild of carrion insects. So flesh flies have a central role as decomposers and recyclers of organic matter, and they constitute an important guild in every ecosystem. A colleague of mine the other day in a TED Talk said that the fly maggots are the original hipsters because they feed only on organic stuff and they are very good at recycling. However, describing and understanding the diversity of these flies is very challenging because these flies look more or less the same, all of them. They are very homogeneous in external morphology. However, the male genitalia is very diverse. So here you can see a collage of pictures of phallic structures with some phalluses having one big opening like the one in the bottom, while other phalluses having 2 or 3 tubes for the sperm exit. So the complexity of these structures, the variation in morphology, and the composite nature of the male genitalia make these structures as very suitable for taxonomic and phylogenetic studies. So we have many questions regarding these three aspects that are in the slide, and our aim is to answer these questions by analyzing the flesh fly diversity into a phylogenetic context. So recently I analyzed male genitalia characters of all genera of the subfamily Sarcophagidae, which is about 2,000 species. Of course, I didn't include them all, but I include all the genera, and we recover a very well-supported tree. However, we still believe that we need to improve our understanding of these structures because they are very complex, and their homologies, as like the identity of these structures, is still very obscure. By using anchor hybrid enrichment, we confirm a pattern of a rapid radiation in the evolution of these flies. In this study, we included all the subfamilies of the Sarcophagidae, and we found very unexpected relationships between them, with Paramacronicinae, a sister to Miltogramminae, although Paramacronicinae is considered as morphologically closer to Sarcophagidae. So there is a strong conflict between molecules and morphology. In this new stage of my research, I will be using ultra-conserved DNA elements, or UCEs, as the data to reconstruct the phylogenetic relationships, both at the genus level in the family Sarcophagidae, and also at the family level within the clade of Oestroidea. So the Oestroidea superfamily include these families here: the blowfly, the botflies, the flesh flies, the woodlouse flies, also the blowflies, and the bristle flies. I think you are very familiar with some of them. So these are some pictures of this ongoing project. When I do field work, I go to hilltops to collect my flies, because flesh flies congregate in these small areas in the top of the mountains to reduce the area, and by doing that, they increase the probability of finding a female to mate. I also get to see very nice landscapes, and at the end of the day, I do the identification, I pin some flies, and some others are stored in a liquid nitrogen tank that I carry using a pickup truck before I send it back to the biorepository here at the Smithsonian. So I've been collecting in very different landscapes and localities here in the States, especially in California and Washington, and also in remote localities in Denmark. By doing this, I am increasing the collections both at the biorepository and also the dry collections here at this museum. The next step of this project is sequencing a flesh fly genome to design the UCE prop set. We expect this UCE prop set to be useful across other fly families. We are going to use transcriptomes to help the genome annotation, and in the end, with the UCE data, we expect to reconstruct a more robust phylogeny of these flies and be able to answer our questions about this group. Thanks to Torsten and Thomas, my collaborators, and also to the GGI for the funding for my project, and also the Department of Entomology, and all of you for being here. Thank you.
So one of your first slides, you showed a picture of a flesh fly hanging out along the margin of a carnivorous plant.
Oh, yes.
So is it about to be eaten, or is it capitalizing on the fact that other things have died there?
Exactly, yeah. They, you know, the line between being a parasite or a saprophagus fly is very fine. So these flies, we don't know if, so she's talking about the picture of the pitcher plant on top, the green one. So flesh flies go to these plants and they eat the animals that are either dying, injured there, or that are already catch and dead there. So the line between being a saprophagus or a parasite is very fine, and we actually don't know if they are true parasites or necrophagous insects. So that's also another question we want to answer through my project.
I'll start with my favorite question. Again, why are you here? And we can maybe start from the other side this time. Why are you interested?
In my flies? Well, I think in the first slide I explained that very well. These flies are very important for forensic entomology, and they are also a very interesting model to study the evolution of the morphology of the male genitalia in insects. And I think they are very nice, very beautiful. Sarcophagids.
Could you please repeat the question?
Sorry, yeah, she asked me about how old are the sarcophagids, I guess in evolutionary times, of course. This family is a very young group. The entire Oystroidea, we actually don't know because we don't have fossils of these flies. We don't have any fossils of tachinids or sarcophagids, calliphorids. We don't have anything from that. The closest fossil we have is from a musid that is not in Oystroidea, but it's all the flies that have calyptures, so it's the calyptrates. And the date that we have so far is 50 million years back. So that's in the Eocene. However, the diversification of the flesh flies apparently has to do something with the Isthmus of Panama. And apparently the closure of the sea, this very ancient sea, or the emergence of the Isthmus of Panama facilitate the migration both from the south, from South America to North America, and also from North America to South America. So apparently the origin is in that area. So they probably are very much younger than we know right now. Yeah.